Hello students, this is part 2 of gene expression in eukaryotes, gene expression regulation in eukaryotes, where I discuss you the regulation through variation in the DNA per se. Now under this heading, I need to discuss few subtopics like the access to the DNA, where I discuss the accessibility of the DNA portions to various transcription factors. We have euchromatin regions, we have heterochromatin regions in the chromosome. The dense area are heterochromatin regions and less dense area are euchromatin regions. It means that the area which is having dense packing of the DNA is called heterochromatin regions of the chromosome. Their accessibility to the transcription factor, to the gene promoter areas are impeded. So those genes are not expressed easily. But in euchromatin, which is loosely packed DNA, the accessibility to the accessibility of the transcription factors to control elements of the gene is easy, and so genes are easily expressed. So we have areas of DNA packed in the euchromatin regions or the heterochromatin regions that decide the level of gene expression. Then next heading under this will be amount of DNA, amount of gene which is available for transcription. Under this heading, you can always talk about gene amplification. Number of times the gene amplify, and that decides the level of transcription. The example I can give you, the treatment by methotrexate, which is dihydrofolate reductase inhibitor. So when methotrexate is given for long duration, this enzyme gene is getting amplified, that results in resistance of methotrexate. So this is dihydrofolate reductase inhibitor, and when chronically it is given for long duration, this dihydrofolate reductase enzyme gene is getting amplified, and that results in more production of dihydrofolate reductase enzyme, abolishing the effect of methotrexate in that patient. So that develops the resistance. Now arrangement of the DNA can be discussed under this heading, arrangement of the DNA, how the exons and introns are arranged, that decides the gene transcription. Then mobile DNA elements, there are certain mobile DNA elements that are called transposons that determine gene expression. So these are the factors which need to be discussed under this heading, where the DNA variation will regulate the gene expression. Access to the DNA, how easily the gene is accessible by the transcription factors. Then amount of gene which is available for transcription, that is gene amplification. Then arrangement of the DNA, how the intron exons are separated. Then mobile DNA elements, there we discuss the transposons. Now you should know one thing, that the histone protein play a very important role in deciding the chromosomal area being euchromatin or heterochromatin. Histone is a basic protein which is wrapped by the DNA. Histone is a positive protein, basic protein. It's having rich amount of arginine and lysine, so it is having positivity. Now, the covalent modification of this histone, either by acetylation or by methylation, changes the ionic charge on this histone protein. For example, when acetylation is there, histone becomes less positive, more negative. Then the nucleosomal structure gets disrupted. That results in euchromatin condensation. And opposite happens when methylation occurs, which increases the positive charge on histone, and nucleosomal structure is favored and heterochromatin condensation is then favored, resulting in gene suppression. So this kind of histone protein modification by acetylation and methylation and its impact on the gene expression is called epigenetics. So in eukaryote, such mechanisms are also involved in regulation of the gene. So in eukaryote, the regulation of gene, I will summarize, can be discussed at the level of transcription, where I discussed your hormone response element, and it's a coordinated regulation because one particular factor is having impact on number of genes. Then mRNA processing, that is post-transcriptional modification, we have number of factors which determine the mRNA stability, mRNA changes in terms of editing, polyadenylation changes, alternative splicing, which is going to have impact on the gene expression. Then regulation through variation in the DNA, where we talk about the chromosomal structure, the condensation of DNA in the chromosome in euchromatin or heterochromatin regions, that in turn is determined by histone appearance, histone covalent status. Then the number of genes will decide the gene expression. Then arrangement of the DNA and the mobile elements of the DNA in turn will, they will decide the eukaryotic gene expression regulation. Thank you very much.